By measuring brain activities of 2 or more people simultaneously, this method can shed light on the neurobiological mechanisms underlying social interactions.
Functional near-infrared spectroscopy, or fNIRS, is well suited for conducting such hyperscanning experiments, because it measures the brain hemodynamic response with a high sampling rate and can be applied in relatively natural settings.
While this protocol shows how to conduct simultaneous fNIRS recordings of adult-child dyads, it can be adapted to different dyadic constellations, such as romantic partners, and to different experimental tasks.
Begin by choosing a cap size that is the same size or slightly larger than the participant's head circumference. Cut 15 holes with a diameter of approximately 15 millimeters each, arranged in a horizontal 3x5 grid, into the forehead area of each of 2 raw EEG caps. Ensure that the holes are spaced 30 millimeters from each other in any direction, that the middle column of holes is located in the center of the forehead. In order to make the caps more comfortable and minimize pressure marks, attach soft foam material at the inner side of the holder grid between the probe sockets and at the edges. Next, mount an empty 3x5 probe holder grid to each of the modified EEG caps, such that the holder grid itself is placed on the inside of the cap and the holder sockets stick in the holes. Then, gently insert the probes into the appropriate holder sockets on the grid such that only the first ridge of each probe is mounted in the socket, which results in one clicking sound. Open the probe set monitor window at the NIRS measurement system and select 2 probe sets arranged